Genes! No, not those. Are segments of DNA that code for the production of proteins. The genes in your cells code for proteins that have specific functions to keep you alive. This is precisely why we deem DNA as the basis of all life. It instructs our bodies how to perform at a cellular level. Since we know this, scientists have been looking into how we can edit genes to create new forms of life.
So that means I can edit genes to turn ordinary turtles into Teenage Mutant Ninja Turtles?
Kind of.
Great, so let's start!
Start what?
I'll be sharing my recipe on how to make a genetically modified organism.
Commonly known as GMOs, these are organisms that have had their genes altered through techniques that you're going to learn about right now.
First, you need to isolate a desired gene. The gene I'm using has a code that tells cells to produce proteins that bulk up turtle muscles, but you can use whichever gene you want. Now, there are many ways to obtain a desired gene. The first way to do this is you can take nucleotides, the most basic stuff that make up DNA, and pair them up. You must know the sequence to line these nucleotides up, since 3 of them code for 1 amino acid. Many amino acids join together to form chains, which then fold to form proteins. So the wrong code can produce the wrong protein, which can have the wrong function, so be careful. Now let's say you don't want to line nucleotides up from scratch. Another way you can obtain a gene is you can take these specialized scissors called restriction enzymes to cut out parts of a DNA molecule that may have the gene that you're looking for. You may even cut out several DNA fragments and join them back up with this specialized glue called ligase. Make sure you use the same exact restriction enzyme scissors when cutting out fragments, because using different ones will cut out non-complementary ends that cannot be joined by ligase. Okay, now that you have your desired gene, it's time to insert this into a plasmid, which is basically just some circular DNA found in bacteria. Cut open the plasmid with the same restriction enzyme scissors we used earlier, so the ends of it are complementary to the ends of our obtained gene. Now glue the gene and the plasmid together, and ta-da! We've just formed something called recombinant DNA. This can now be inserted into a vector, which is basically a carrier. An example is bacteria. Bacteria and the recombinant DNA are mixed together in a solution of calcium ions so the bacteria can take up the plasmid, and when they do, we've just formed genetically modified bacteria! Now this bacteria can produce proteins that bulk up turtle muscles, and I can get a step closer to forming my Teenage Mutant Ninja Turtles.
But in reality, it's a lot of work to create an entire organism. These are just the key principles of genetic technology, and yet we've already done so much with this. Hormones have been created to treat diseases like diabetes, illnesses have been diagnosed, vaccines have been developed, and even climate change is being reduced. So what truly makes a breakthrough is how we use such technology. So maybe next time I'll be doing something a lot more significant than just making some Teenage Mutant Ninja Turtles.